Sort of as usual, I don't really have anything to say or contribute, so I'm here to answer any questions you guys have about anything. Not even done with the exam. I almost got it done today, but I'll have to get finished over the weekend. Yeah. Okay. Yeah. No, that's fine. I know it. I've been asked about this one several times. Yeah, so this question's definitely been bothering people. So we do sort of present as if there's like an upper motor neuron that sends a signal from the primary motor cortex down to the lower motor neuron, and that carries the skeletal muscle. And that's not really wrong per se, but there are, maybe vast oversimplification is kind of strong, but there are maybe two major points that I'd point out where this isn't kind of a complete story. So these are both coming out of this chapter. So one kind of has to do with like this monkey joystick experiment. So in this experiment, what they demonstrated is, so they sort of caused the monkey to push its arm in one direction or another, because if it did so, it got a treat. And these are fairly simplistic actions. But what they did is that they recorded from a whole bunch of neurons, upper motor neurons, that are up in their primary motor cortex. So these are populations of M1, stands for primary motor cortex. And so what they found is that when the monkey is pushing the joystick to the left, there was this whole population of neurons that would light up. It's not like there were a handful of them. And some of them, and not all of them, are directly always involved in movements to the left. So a lot of these neurons, like these guys, for example, their main direction of movement, these ones that are represented by these individual lines, is actually to push the joystick up at the diagonal. But when you push to the left, they also seem to be involved. And there are even some of these same neurons that get involved in pushes to the left are also involved in pushes up or pushes down. So the argument here is that for any given action, even a pretty simple action like move joystick this way or this way, there's this whole population of upper motor neurons that are all getting excited. And they're all sending signals down to a big population of lower motor neurons. And then the lower motor neurons kind of interpret all those signals, and then that determines which muscles actually get excited. So it's not like there's like one upper motor neuron that leads to one lower motor neuron. There's this population of upper motor neurons, even for simple actions, leading to a population of lower motor neurons. So that's one thing. The other thing I pointed out, and there's this picture, there's another one that's kind of like this, probably up here. Yeah, we'll use this one for this, in this case. So what this is trying to represent is this motor loop. And in part, it's, I think a lot of this figure is focused on this part of the motor loop. So this is this circuit that goes through the basal ganglia, starting in the prefrontal cortex, and you go through the basal ganglia to the thalamus and back to the supplementary motor area. But the thing I want to emphasize for this purpose, so we were saying in that question that it's upper motor neurons in the primary motor cortex. That's these guys over here in area 4, and they definitely contribute axons that go down the spinal cord in that cortical spinal tract. But I think what this figure points out is that there's also actually axons coming out of area 6. And area 6 includes the premotor area and the supplementary motor area. And so, like a lot of times, I think we tend to think of area 6 telling area 4 what to do, and area 4 then sends signals down. And that does happen, but we also have axons coming straight out of area 6, going straight down the spinal cord. So it's not just primary motor cortex that has upper motor neurons. It's these other sort of association areas that are also sending signals directly down the spinal cord too. So I think those are like the two main things I was thinking of when I wrote that question. Like I said, I've been asked that one at least a couple times this week. I would never put that as like a question on an exam, but study guide, it's kind of, I don't know, anything goes. What else can we do? Mm-hmm. So yeah, the lateral corticospinal tract, I think you can think of as like our main intentional motor pathway. So it's like the main motor pathway that we're directly conscious of, is how I think of it. So if you think about any given action, so maybe my common example is if there's a thing sitting on the table, and so the motor action for me is to walk up to it and pick the thing up. So like, I may be, let's do it again. So I may kind of do this and reach and pick it up. So like I'm mostly focusing on what's going on with my arm and my hand, and I'm gripping the thing. But what I may be less cognizant of is, other things going on in terms of like, my leg muscles and my core muscles and such that are helping me to maintain balance as I lean forward to pick up this thing. But the corticospinal tract is kind of like running these distal limb muscles. That's what it mostly does. So when we were just talking about those upper motor neurons, those upper motor neurons in the primary motor cortex, area 4, as well as I guess also in area 6. So they're starting up here in this motor cortex area. And they're sending axons that travel down through the brain stem. So I'm going through midbrain, I'm going down through the medulla. They decussate down here in the medulla, and then they're traveling all the way along the lateral corticospinal tract, which is this little blue dot, or cylinder, I guess it really would be, that's traveling through the lateral column down to a particular level of spinal cord where it will exit, and then along with lots of other axons that are kind of going the same direction, it'll stimulate the lower motor neurons that are down here. So that's the lateral corticospinal tract. It's this pathway that's traveling directly from your primary motor cortex down to the spinal cord to the levels where we've got lower motor neurons found down here in the ventral horn of the gray matter that are going to be excited, and then those in turn carry the signals out to the skeletal muscle. So in a way, it's kind of like the classic main motor pathway through brain, through spinal cord. It's certainly the one that we're sort of most conscious of in terms of the movements that we're most conscious of. This is what is sort of driving those movements. The other tracts, like the vestibulospinal tract, for example, or especially these like reticulospinal tracts, these guys are kind of operating they're operating sets of skeletal muscles, and they're skeletal muscles we have voluntary control over. But these are kind of operating in the background. So as I kind of lean forward to pick the thing up, they're the things that are helping me to like maintain the tension and appropriate contractions in my core muscles and my leg muscles to keep me from falling over. So they're operating, for example, these anti-gravity reflexes in my legs that help me to not fall onto the ground and maintain balance. The same is true, like the vestibulospinal tracts are helping me to maintain balance a little bit of a different fashion. This is based on the signals that my vestibular system is receiving, and then correcting limb motions and neck and core muscles to help maintain balance based on based on that vestibular input. So that's the idea. Yeah, it's almost like these things are, yeah, it is easy to overthink it. You can almost think of these other systems as kind of like the vestibulospinal tracts, tectospinal tract, reticulospinal tract. They're almost more like reflex-based, so we're kind of less directly in control of them. Whereas corticospinal tract, this is much more like the voluntary motor pathway. I don't remember. Not obviously. Yeah. I mean, the way it's drawn here, I guess they're kind of showing it going to both sides. But but it's not like all the fibers are decussating. Yeah. Mm-hmm. Yeah. For depression, is it? It's terrible. Yeah. Okay, so what is the monoamine hypothesis? So this is coming from this discussion of affective disorders. Let me make sure I'm right. Yep. So we're looking at major depression. We're looking at bipolar disorder. And the way I approach this is what your book does too, is kind of stemming, if we're trying to understand what's causing affective disorders, what's causing disorders of mood. One way one can do this is that based on other pharmaceutical studies, when people have been giving people drugs and it's caused a change in mood, we could then look and see if that's associated with some sort of common mechanism. So we talked about this reserpine, which is a blood pressure drug that it turns out does affect catecholamines, which is like acetylcholine, and serotonin, and how well those load into synaptic vesicles. And if you give people this stuff, it can sometimes cause severe depression. There's these tuberculosis drugs that interfere with an enzyme, monoamine oxidase, that destroys these same kinds of neurotransmitters, catecholamines, acetylcholine, and serotonin. And it also affects mood. So if you get rid of, how does this work? So if you get rid of these, so normally this enzyme does get rid of these neurotransmitters, but here we're blocking the enzyme, so we have more of those neurotransmitters and it causes elevation of mood. So it's like the opposite of this thing with the same kind of mechanism. And then this other one, imipramine, was an antidepressant where it turns out that it helps to reduce uptake of serotonin and norepinephrine. Serotonin, at least, is common across all these three. Norepinephrine is also kind of in the same category. So catecholamines are like acetylcholine, serotonin, norepinephrine, all these things are monoamines. So if we go back to like chapter 5 or 6, where we covered neurotransmitters, we had monoamines as like one major category of neurotransmitters. We had amino acids, and then we had peptides. So all these guys that are listed on here, these are all monoamines. So that's where this monoamine hypothesis came from. This idea that if you have inadequate levels of monoamines, that might then lead to depression. And so some of the pharmaceutical treatments for depression are about trying to increase the numbers of these neurotransmitters. So this is where like these those serotonin selective reuptake inhibitors are helping us to elevate our levels of serotonin, and then that in turn may help to mitigate the depression. It seems to work sometimes. So that's the idea. The other treatment I'll just mention, since we're here. The other treatment we talked about, where'd it go? Oh, I'm on a different topic. Never mind, I'm going to stop talking. Cut that. We'll cut it out of the recording. Getting myself confused. Yep. Yeah, so that there's this interaction. There's some kind of involvement. So as far as HPA axis, mostly we talked about HPA axis with anxiety, but with depression, there is this effect that can happen where normally what moderates cortisol is you have this feedback system where if you have too much cortisol, it stimulates glucocorticoid receptors in the hippocampus, which then reduces HPA output. But you can, if you have sort of chronic cortisol over an extended period of time, that can actually damage your hippocampus and kind of affect that feedback loop such that your hippocampus is no longer able to then moderate the amount of cortisol that's being produced by inhibiting HPA axis. And the connection with the monoamine stuff is that, and this is where this comes in, that's also, there was the homework paper that for this chapter that I had you guys read, is that Serotonin, in particular, one of the monoamines, tends to increase the number of glucocorticoid receptors in the hippocampus. And so if you don't have enough glucocorticoid receptors, because you don't have enough serotonin, then that in turn could lead to not enough inhibition of the HPA axis, which then could lead to too much cortisol, which could lead to further damage of the hippocampus. And so then you have kind of this runaway system. So there is certainly an implication of HPA axis in depression, and there's also implication of it in anxiety disorders too. And so for that reason, some of those SSRI drugs are used to treat both anxiety and depression. So you're elevating serotonin levels by not by blocking the reuptake inhibitor, well, by blocking the reuptake protein with an inhibitor. And so that in turn may lead to more glucocorticoid receptors because there's more serotonin, which will allow you to moderate the HPA axis better. So there's something going on there. That's the idea. All right. What else would you guys like to do? Mm-hmm. Yeah, I'm trying to remember what that was. Nope, wrong chapter. Yeah, let's see. Where did we talk about that? That's one of these guys. So chlorpromazine, you can kind of see it. It's really small, but it's this guy. So this is one of these neuroleptic drugs. And what this figure is showing is that it does interact with the D2 receptor. That's the dopamine 2 receptor, although it doesn't have very high affinity. So this is a weird graph, but they have the low affinity is a bigger number than the high affinity is. But again, one of the things it seems to do is it interacts with this dopamine receptor. Let me flip over because I think I had, yeah. So the mechanism, I mean, so if the argument with schizophrenia is that I've even seen cases, in fact, somebody wrote a paper or read a paper and did a paper summary on I don't remember who it was because I was grading them all anonymously, but they kind of describe there being like this Parkinson's, schizophrenia spectrum because they're all related to dopamine transmission. And so one of the arguments for what goes on with schizophrenia is you just have too much dopamine around. And if there's too much dopamine around, it's causing this problem. Whereas Parkinson's was not enough dopamine being around. So what this drug, chlorpromazine, along with most of these other neuroleptic drugs do, is they bind to this D2 receptor. D2 receptor is just a receptor for dopamine, and therefore, even though you've got a lot of dopamine around, there are fewer receptors available because they've got this drug basically masking them and blocking them. And so as a result, you don't have as much dopamine neurotransmission, and so you have less of the symptoms. So that's really, I mean, it's a dopamine antagonist would be the short answer. And I guess that also bridges into that homework question, because the homework question was, I don't know if it specifically referenced one drug, but a lot of these neuroleptic drugs lead to movement disorders. And the reason for that is that you're blocking these dopamine receptors, and those dopamine receptors, if they're blocked, if too many of them are blocked, then you could have basically, you're sort of inducing Parkinson's on a person, at least on a temporary basis, because you're blocking their dopamine transmission, and therefore they can't start the motor loop, just like we talked about back in chapter 14 with Parkinson's. I'll go there. Yep, there it is. So normally dopamine does this. It helps to stimulate the putamen such that the putamen is sort of partially stimulated and ready to respond to frontal cortex neurons. So if you get rid of dopamine transmission, either by, in the case of Parkinson's, these neurons are dying, or if you're taking lots of neuroleptic drugs, they're blocking the receptors for dopamine, and therefore you're sort of blocking the stimulation. That can lead then to the movement disorders they end up looking a lot like Parkinson's. So sort of a pick your poison problem. Right, what else? Mm-hmm. 13. So this one, what are two specific molecular mechanisms that are thought to be responsible for the debilitating effects of amyotrophic lateral sclerosis? Yeah, so this came up on a homework. I'm going to pull that up just so we have something to point at. In fact, I've got my answers written here. So there's ALS. So your book had a little box that I asked you guys to read for this one, and basically it talked about two things. One was excitotoxicity. So the argument here is that if you have too much glutamate transmission, so glutamate's a important stimulatory neurotransmitter. It's also amino acid. But it's if there is too much glutamate neurotransmission, this seems to lead to an overstimulation that leads to cell death of these motor neurons. So you can basically overstimulate them, and then that overstimulation seems to lead to some of this alpha motor neuron cell death that happens in ALS and causes you to lose your motor control. There was some indication there might be in some cases like a defective glutamate transporter, so there can be some genetic basis to this if you have a poor or a defective glutamate transporter, and as a result it doesn't reuptake glutamate like it should, so glutamate will linger in the synapse and therefore overstimulate your neurons. So that was basically item number one. The other mechanism that was discussed was oxidative damage, and I think there's some indication that the excitotoxicity can lead to oxidative damage, or the excitation can lead to oxidative damage, which can lead to some of the toxicity. But independent of glutamate, there's also indications that if you have damage to an enzyme called superoxide dismutase, then that can lead to an excess of oxidative damage, and that oxidative damage then can lead to the cell death of these cells, or malfunction of glial cells, or loss of neurons, and these kinds of things. So, so again, it's sort of excitotoxicity by glutamate, and then oxidative damage, maybe because you don't have effective superoxide dismutase. Normally what superoxide dismutase does is it helps to get rid of excess superoxide molecules, which are basically oxygens that have extra electrons attached to them. And they're super reactive and just blow apart biological molecules. I don't think if there are any other big movement disorders. I mean, so we definitely talked about like Parkinson's, we talked about Huntington's, Ballism, in this section. I think earlier in the semester we hit like multiple sclerosis. What was the other one? Myasthenia gravis we did earlier on. It's probably most of the big ones. What else can we do? Mm-hmm. Chapter 14, question 13. Oh, good. Yep. So this one about basal ganglia, how they do that. Yeah. So this is that story about the motor loop. We were sort of just there, but we didn't actually do the story. And then there is this question: if the putamen has an inhibitory effect on the globus pallidus, how does that in turn result in excitatory signals going back up to the motor cortex? So it's, I always say the thing about this one is that you've got sort of like inhibition of inhibitors, which is always just takes me a little bit of extra time to get my head around. But to walk through it, in order to start a motor action, we have to send signals, usually from our frontal cortex, which is sort of like, this is our conscious decision-making, we're sort of willing ourselves to make some particular motion. So from the frontal cortex, we'll send signals over to the putamen, which is getting stimulation, sort of like a tonic, consistent stimulation from the substantia nigra of dopamine. And so the dopamine kind of partially stimulates these neurons so that when they do get signals from the frontal cortex, they are ready to fire, and they do get excited. Their job, as the question says, is that the putamen cells inhibit the globus pallidus cells. So we are turning off the globus pallidus. The job of the globus pallidus is to inhibit the thalamus up here. This is this ventrolateral nucleus of the thalamus. So what we are doing basically is the putamen is turning off the inhibition that otherwise would be coming from the globus pallidus. So we're shutting down this neuron, which means it is no longer inhibiting the thalamus. So this is called disinhibition. It's a good word. And so by disinhibiting the thalamus is now sort of freed up. It's like you're taking the brake off of it. And so it will be able to start firing and sending signals up to the supplementary motor area. And it's these signals going up to the supplementary motor area that allow you to kind of start an action, to get that action going. So we do start with like stimulation going into this system, and then we end up with signals coming, excitatory signals coming out of the system. But the sort of steps to doing so is we have to inhibit an inhibitor to free up the thalamus so it can start firing. That's sort of the story with it. I don't know, sometimes I find it useful even to kind of sketch it out. Not in like full anatomical glory, but you know, you could have almost just in a line, you know. Frontal cortex, putamen, globus pallidus, thalamus, SMA, and just try to remember what each of those neurons is doing. And the key thing that we have to do is we need to shut down globus pallidus neurons so that we can free up our thalamus neurons. That's the key to making the whole system operate. I think the only other thing we talked about that's related to this is normally the thing, so the reason globus pallidus neurons need to be shut down is that otherwise they're receiving sort of constant stimulation from the subthalamus, which is this little structure over here. So the subthalamus is constantly stimulating the globus pallidus. So without any signals coming from the putamen, if that's not there, then you basically have subthalamus stimulating globus pallidus, which is shutting down the SMA, shutting down the thalamus neurons. And these thalamus neurons, therefore, can't excite the SMA. And that's preventing you from doing any kind of unwanted actions. So if you have ballism, what can happen is these neurons start to die, which means you lose that excitation of globus pallidus, which you start then to free up the thalamus kind of when you don't want to, and all of a sudden you have these kind of, kind of flailing kinds of actions that happen in embolism because you are no longer suppressing motion like you normally would be. So I think sort of between Parkinson's, which is losing your substantia nigra, and embolism, which is losing your subthalamus, it gives you these sort of two, you're manipulating those two inputs into the system, and it causes these different effects. Just getting kind of loud out there. So most of that stuff on the ballism and such, that all came in the homework for chapter 14. Oops. Oh. Yeah, it's like this stuff. Mm-hmm. Yeah, similar kind of format. So mostly multiple choice. Probably a little bit longer than usual, but not like, I don't think it'll be double as long. And I would guess probably most of the extra length would be in the multiple choice section. I haven't finished it yet, but it's looking how it's going to be. But there'll probably be a little bit of a kind of mixed random response section. There'll be a little bit of a fill in blank section. So same kind of format. Shouldn't be anything that, the intention is never to like totally throw you. Doesn't mean I don't successfully still manage to do it, but I don't intend to do it. Other questions? I think if there's anything like big, but I don't know. Anything else? All right. We can just kind of call it then. That's fine. I may stick around a few minutes in case somebody comes in from an exam, but that sounds good. Well, have good weekends. Do you guys have Saturday exams? Lucky you guys. Yeah, I didn't get any this year either, but I've only had like one or two in my career here.